Welcome to the second video in the bacterial metabolism unit. In this video, we're going to take pyruvate that we talked about in the last video and continue it through the metabolic processes, comparing the differences between fermentation and respiration. Throughout this video, we're going to talk about processes that you've probably learned about in other classes. However, I'm going to stress repeatedly that what you learn in other classes isn't necessarily the full picture. In bacteria, these processes can happen in the presence or absence of oxygen, which tends to be a little bit different than what we learn in other classes. And so often you're going to hear me say in this video, you might have learned this way, but now we're going to have to break that thinking a little bit and start looking at it in a larger picture because bacteria can do much more diverse things than your cells can. All right, let's take a look. Remember from the last video that in glycolysis, a molecule of glucose is broken down to 2 molecules of pyruvate. In this process, we also produce a net yield of 2 ATP and 2 NADH. Remember that those 2 NADH are formed because 2 NAD plus molecules come in to pick up the electrons as glucose is being oxidized. In order to keep glycolysis going, we need to ensure that we have a continuous supply of NAD+, because if it runs out, this process is going to stop. This means that NADH must be able to drop off its carried electrons somewhere in order to reform the NAD+ that is required. Cells can accomplish this in 2 ways, by funneling pyruvate either into fermentation pathways or into respiration. If it follows to fermentation, no more energy will be produced, but NAD+ will be regenerated. If it funnels towards respiration, then the molecule will be further broken down and more energy will be released. The determining factor for which branch is used is based on a functioning electron transport chain. So if there is an electron transport chain that is running in the cell, pyruvate will funnel towards respiration. However, if there is no electron transport chain, then fermentation will be used instead. Be sure to note here that I stated respiration will proceed if there's a working electron transport chain. I did not mention oxygen. In most other classes, you learn that respiration happens in the presence of oxygen, while fermentation happens in its absence. That is only true for organisms like yourself, who are obligate aerobes. It's not necessarily true for many species of bacteria. First, we'll look at the fermentation branch, because that's the shorter of the 2 options. Fermentation does not require oxygen or any other inorganic electron acceptors that may be powering an electron transport chain. Instead, it's going to take the pyruvate that had been produced from glucose, and it's going to convert it into some other product. This other product differs between species, but in all instances, it is actually a waste product. And so it's not used by the cell, but instead can be released from the cell. And in fact, if too much waste product builds up, it can be detrimental to the cell itself. Since no other energy is produced during fermentation, the bacteria are relying on an incomplete digestion of the sugar, meaning that they have to consume large amounts of sugar to obtain the ATP they need to survive. While this can be difficult, there are some microbes that rely solely on fermentation for their energy generation. While this other product of fermentation is a waste product, the key here is that to convert pyruvate to the other product, we require NADH to come in and drop off its electrons. And so pyruvate is actually gaining electrons in this process. It's being reduced, and along the way, NADH is being recycled back to NAD+. That NAD+ can then funnel back to glycolysis so that we can keep breaking down glucose and continuing to produce those 2 ATP molecules as energy for the cell. From our perspective, we actually rely on fermenting microbes to produce a number of products for us and to diagnose certain species. Fermentation was first used by humans as a food preservation method. For example, sauerkraut, or fermented cabbage, will last much longer than the cabbage itself. Similarly, fermented milk products, like yogurt, will last longer than the milk itself. Fermenting microbes are used for beer and wine industry. They're used in chocolate and coffee production, soy sauce, and many other products. For diagnostics, fermentation products are typically acids, which lower the pH of their environment. This lower pH is why fermentation can be used to preserve foods. But the change in pH can also be detected by changes in media color, allowing simple diagnostic methods, such as the McConkey agar plate we saw when we were discussing the bacterial growth media. This slide illustrates the variety of fermentation products that exist in microbes. You do not need to memorize all these pathways, but you should note that NADH, shown in the blue, is being used in every single one of the reactions. So we're recycling NAD+, which is the entire purpose of fermentation. Organisms that are doing fermentation are only extracting 2 ATP from each molecule of glucose during glycolysis. There's still a lot of potential energy in the remaining pyruvate molecules, and that energy is released if pyruvate can continue down the respiration path instead of the fermentation path. This is going to allow those molecules to be completely oxidized to carbon dioxide, extracting more energy from them. There are 2 forms of respiration, however: aerobic, which requires the presence of oxygen, and anaerobic, which can occur in the absence of oxygen, but instead requires the presence of a different inorganic molecule. In past classes, you've learned about aerobic respiration, which typically will produce between 32 and 38 molecules of ATP. But often you do not learn about anaerobic respiration. In anaerobic respiration, many of the pathways that we've already learned about still occur, except the electron transport chain is going to use something other than oxygen as the terminal electron acceptor. This means that it's not going to produce quite as much ATP as in aerobic respiration. Typically, it's going to produce a little bit less, but it does produce more than fermentation. You're not going to have to calculate the amount of ATP that's produced during anaerobic respiration for this class, because the number is different depending on the electron acceptor that's actually used. So we're going to walk through the pathways of respiration and highlight the key difference between aerobic and anaerobic respiration processes. When pyruvate moves into respiration instead of fermentation, it is first oxidized to a molecule called acetyl-CoA. This is sometimes called the transition step or the bridge reaction. Alternatively, you will sometimes see it written as part of the TCA or the citric acid cycle, but it's really its own separate step. In this step, pyruvate is going to be converted to acetyl-CoA through an oxidation reaction. In that process, it produces a molecule of NADH. You'll notice that pyruvate is a 3-carbon sugar, while acetyl-CoA only has 2 carbons. The 3rd carbon from pyruvate has been released as carbon dioxide, a waste product. It's important to note that this is the chemical reaction for pyruvate oxidation. However, if you remember, during glycolysis, 1 molecule of glucose actually produced 2 molecules of pyruvate. What that means is if we're talking about metabolizing a molecule of glucose, then pyruvate oxidation actually happens twice, once for each of the pyruvate, leading to instead the production of 2 molecules of acetyl-CoA and 2 NADH electron carriers. We also would lose 2 carbon dioxide waste products in the process. The next step in respiration is the TCA cycle, which is also called the citric acid cycle or the Krebs cycle. There are 3 different names, all for the same thing. In the TCA cycle, acetyl-CoA is further oxidized to carbon dioxide. In that process, it's going to produce 3 molecules of NADH, 1 molecule of FADH2, and a molecule of ATP or GTP. GTP is just a different nucleotide triphosphate. While not as commonly used as ATP, it's also an energy equivalent for the cell. Again, remember that if we're talking about metabolizing a molecule of glucose, Then there were 2 pyruvates, which led to the production of 2 acetyl-CoA, which means that this whole citric acid cycle or TCA cycle would happen twice. So from a molecule of glucose, we would actually get 6 molecules of NADH, 2 molecules of FADH2, and 2 molecules of ATP. Along the way, we would be releasing 4 molecules of carbon dioxide. It's important to note here that we're talking about bacterial metabolism. You have learned these pathways in past classes. However, you learned them in relation to eukaryotic cells. In a eukaryote, these reactions are occurring in the mitochondria of the cell. But in bacteria, there are no mitochondria. All the reactions we have been talking about are happening in the cytoplasm of the cell. The TCA cycle is also a funnel point for other metabolites to enter respiration, as well as a point to funnel products out to go down other pathways. This image highlights how the TCA cycle is used not just for energy generation, but also in anabolic pathways for biosynthesis of various compounds. You'll notice that various products in the cycle can, instead of traveling around the cycle, leave and be used to make molecules, such as citrate being able to leave to make fatty acids, or alpha-ketoglutarate being used to make glutamate, which is necessary for certain amino acids and also to produce our nucleotides. If a molecule is funneled out of the cycle, then the remaining steps after it do not occur during that turn, and the energy products will not be generated. In the alpha-ketoglutarate example, the 2 remaining NADH here and here, along with the molecule of ATP, would not be produced because alpha-ketoglutarate would have actually left the cycle instead of continuing on through. The decision of a molecule to continue through the TCA cycle or be funneled into biosynthesis is based on enzyme regulation and what the cell needs. If there's plenty of ATP, then the cell will synthesize new molecules instead. But if ATP is needed, the TCA cycle will continue. We're not really going to discuss the details of all these anabolic pathways in this class. I just want you to understand that they exist and that they're all interconnected. Okay, so to summarize, by the end of the TCA cycle, all of the carbon in the original glucose has been released to carbon dioxide. A little bit of ATP has been produced through what we call substrate-level phosphorylation, and the remaining energy is currently in the form of NADH electron carriers. At this point, glucose started in glycolysis, was converted to 2 molecules of pyruvate, which led to a net yield of 2 ATP and also produced 2 molecules of NADH. The pyruvate then continued on into pyruvate oxidation, where they were converted to 2 molecules of acetyl-CoA, and in that process we made another 2 molecules of NADH. The acetyl-CoA then funneled into the TCA cycle, where it combined with a molecule called oxaloacetate to form citrate, which then continued around the cycle. In this process, we made another 6 NADH, 2 FADH2, and 2 molecules of ATP. If we add all of those up, that means at this point we have made 4 ATP through substrate-level phosphorylation, and we have 10 NADH and 2 FADH2, which function as electron carriers and are going to continue on to the electron transport chain. The electron transport chain, or electron transport system, is the last stage of respiration. It consists of a series of membrane-bound proteins that all have an increasing affinity for electrons as you go down the chain. This means that each protein in the chain actually wants to bind the electrons a little bit more than the protein that was before it. So it's going to steal the electrons from it and pass the electrons from one protein to another. As these electrons get passed, they're going to release a little bit of energy, and that energy is going to power the transport of hydrogen ions out of the cell. It's important to remember that there are no mitochondria here. The proteins of the electron transport chain are actually embedded in the bacterial cell membrane, not in a separate compartment. So in order to continue passing electrons, the electrons are going to be removed at the end of the protein chain by an inorganic molecule called the terminal electron acceptor. This is the only conceptual difference between aerobic respiration and anaerobic respiration. In aerobic respiration, that terminal electron acceptor is oxygen, while in anaerobic respiration, it's something else. Species often use things like sulfur. Species often use things like sulfur, or others like nitrate or sulfate. The overall purpose of the electron transport system is to generate the proton motive force of hydrogen ions. This proton gradient can then be used to power ATP synthase enzymes to make ATP through a process called oxidative phosphorylation. Next slide, we're going to take a look at what this actually looks like. Okay, so here we have our bacterial cell. So this is the normal cell membrane. And embedded in that cell membrane are the proteins of the electron transport chain. Also embedded in the membrane is a protein for your ATP synthase. This process starts when your NADH that has already been made in those previous steps of respiration comes along and drops off electrons to the electron transport chain. They drop them off at this first complex 1, so the first protein in the chain. And when those electrons are dropped off, NADH is now oxidized back to NAD+, which remember can now go back to things like glycolysis or the TCA cycle to pick up more electrons and come back and start the process again. Once the protein has the electron, those electrons are then passed all the way down the chain because each protein in line wants those electrons more than the first one, so it pulls them away. So here we have step 1, electrons are dropped off. Step 2, electrons are transferred. And then during that transfer of electrons, the energy that's being released from the electrons is going to be used to power the pumping of protons out of the cell. This creates a proton gradient, what we also call the proton motive force, where we now have a high concentration of protons outside the cell compared to a lower concentration in the cytoplasm. Anything that has high concentration on one side and low concentration another will like to diffuse down its gradient. And so those protons will diffuse through ATP synthase, and that's going to power ATP synthase to take ADP plus inorganic phosphate and combine them to make ATP. So step 3 of the electron transport chain was the pumping of the hydrogen ions to create this proton motive force. Now the last step is that this electron that's here sitting in this last protein needs to be removed from the electron transport chain. These proteins can only hold so many electrons, so if this one stays here, then we can't pass any more down and the whole chain gets backed up. This is where the difference between aerobic and anaerobic respiration comes in. If this is aerobic respiration, then what is going to pick up the electrons at the end is oxygen. And that's going to form water, which is a byproduct of respiration when you're dealing with aerobic respiration and oxygen. However, in anaerobic respiration, those electrons still need to be removed from the chain. Everything else is the same, but those electrons get picked up by something else. So for this example, sulfur, forming instead of water, hydrogen sulfide as a waste product. The waste products of anaerobic respiration completely depend on what molecule it is that picks up the electrons at the end of the electron transport chain. If you remember, we also formed two molecules of FADH2. FADH2 will also contribute to this process by dropping electrons off, only it drops them off at this second complex instead of the first. All this means for the cell is that a little bit less proton gradient is made because we skip the first complex, and so we don't get to pump those protons. As you're looking at these, for every one molecule of NADH that drops its electrons off, we make about 3 molecules of ATP, whereas for every one molecule of FADH2, we make about 2 molecules of ATP. So if you remember from the last slide when we summarized everything, we had originally had 10 molecules of NADH. When those all drop their electrons off, they're going to lead to 30 ATP. We also had 2 FADH2, which will lead to another 4 ATP. When you combine the 30 ATP plus the 4 ATP here, plus the 4 ATP that were made through substrate level phosphorylation, you get the total 38 that you often see recorded in textbooks. This slide summarizes all the steps of aerobic cellular respiration, showing where the 38 total ATP would be generated. Remember that the number of ATP produced by ATP synthase is directly tied to the amount of proton gradient that was generated to power it. In anaerobic respiration, the electron transport chain doesn't generate as much of a gradient due to the differences in the terminal electron acceptor, so that value is a little bit lower for anaerobic respiration. This slide is included because it's a good summary slide that can help you study for the exams. I've highlighted the key difference for anaerobic respiration compared to aerobic respiration. This table gives some examples of organisms that use anaerobic electron transport chains and what their products are when they do. For example, Pseudomonas aeruginosa, which is normally a soil bacterium but can also be an opportunistic pathogen, especially in cystic fibrosis patients, are able to use nitrates as their terminal electron acceptor. When those pick up electrons, it's going to form nitrites. If this organism did not have nitrate, it would not be able to run the electron transport chain, and thus it would either die or it would have to rely on fermentation, depending on its other metabolic capabilities. I highlight this to make sure everyone realizes that not every organism will die if you take away oxygen, but instead they might be able to rely on other molecules the same way you rely on oxygen. Facultative anaerobes, which can use oxygen if it's present, but can switch to anaerobic metabolisms if it's absent, often have multiple electron transport systems, one that uses oxygen as a terminal electron acceptor if it's present, and another that uses something completely different as the acceptor if oxygen is absent. Alternatively, some facultative anaerobes do rely on fermentation in the absence of oxygen instead of performing anaerobic respiration. An important thing to note is that in bacteria, the proton gradient created by the electron transport system is not just used to power ATP synthase. In eukaryotes, that gradient is trapped within the mitochondria, and it's used exclusively by the ATP synthase within that mitochondria. But in bacteria, the proton gradient is generated outside the cell and can be used by any number of cell membrane proteins. While much of it is going to power ATP synthase, seen here, some of it is also commonly used in secondary active transport to either bring other molecules into the cell or to release waste products out of the cell. Additionally, it's the proton gradient that powers the motor that causes the flagellar rotation and movement of the flagella itself. These other uses of the proton motive force are one reason that the mathematical yield of ATP production is never actually met. So when you calculate that 38 ATP for cellular respiration, that's never actually met by the cell because the proton gradient is used for multiple other things, which means it doesn't all funnel through ATP synthase. And we also talked about how sometimes products are funneled off into different pathways and don't actually continue all the way through respiration. For both of these reasons, that mathematical yield is never actually seen inside the cell.